Hello everyone, welcome to CAPSA. Today's topic is regulation of gastric acid secretion, or what are the pathways involved in the regulation of gastric acid secretion. So there are mainly four pathways which are involved in the regulation of gastric acid secretion. First pathway is endocrine pathway, where gastrin is involved. Second pathway is paracrine pathway, where histamine and somatostatin are involved. Third one is neural pathway. Acetylcholine is involved in this pathway. And fourth one is autocrine pathway. And in this pathway, transforming growth factor alpha is involved. Now we will understand each pathway very clearly. Okay. So first pathway, that is endocrine pathway, which is controlled by gastrin. Okay. So where there is a food into the stomach, it will stimulate the gastrin release from the G cells, and thus gastrin then binds with the CCK-B receptor or cholecystokinin B receptor on the basal membrane of the parietal cells to induce the expression of potassium hydrogen ATPase pump, and thus increase in the expression and activity of potassium hydrogen ATPase results in the increased secretion of H plus ion and acid formation occurs. So there is a food particle in the stomach causes gastrin release from the G cells. This gastrin then binds with the CCK-B receptor or cholecystokinin B receptor. As a result, induce in the expression of hydrogen potassium or potassium hydrogen ATPase. As a result, increase in the H plus ion secretion, thus gastric acid formation and secretion occurs. So, if there is absence of food or there is no food in the stomach, lumen of the stomach, obviously that will not stimulate the gastrin release from the G cells. As a result, there will be no binding with the gastrin and CCK-B receptor. So there will be no expression in the hydrogen potassium ATPase pump. So less secretion or no secretion in the hydrogen ion. As a result, gastric acid formation or secretion decreases or stops. So absence of food causes no release of gastrin. As a result, decrease in the H plus ion concentration and gastric acid formation or secretion stops. Okay. Now we will understand about the second pathway, that is paracrine pathway, where histamine and somatostatins are involved. Now we will discuss about the first type, that is histamine, how histamine is involved. So release in the gastrin causes increase in the mRNA expression, which accelerates the synthesis and release of histamine, which then binds with the H2 receptor of the parietal cells and stimulates the acid secretion. So if there is gastrin release, that will obviously increase in the mRNA expression, which then causes the increase in the histamine synthesis and the release. Then this histamine binds with the H2 receptor, which is present in the parietal cells, and it stimulates the parietal cell activity. So as a result, obviously if there is increase in the stimulation of the parietal cells, obviously there will be increase in the HCl secretion. So if there is no release in the gastrin, obviously there will be no mRNA secretion. So less histamine release, as a result HCl secretion decreases. And this is the first pathway which is involved, where is histamine is involved. The second pathway is mediated by somatostatin, which is a potent inhibitor of gastrin release, and secretion of somatostatin is regulated by cholinergic transmission pathway. Okay. So if there is increase in the somatostatin release, obviously that will causes decrease in the gastrin release. And if there is a decrease in the somatostatin release, that will increases the gastrin release, which will obviously affect the histamine release from the ECL cells. Okay. Now we will understand that the third pathway, that is neural pathway. So if there is a food particle, food, taste, thoughts, this kind of stimulus produces, it will causes the activation of the vagus nerve. Okay. Which is due to food, taste, thought, this kind of things arises a stimulus which will causes the vagus nerve to activate it, or cholinergic nerve to activate it. Okay. That will release then acetylcholine neurotransmitter, and this neurotransmitter binds with the M type 3 receptor present in the parietal cells. As a result, stimulation of G cells, parietal cells, ECL cells occur. So if there is no stimulus produces due to food, taste, thoughts, this kind of things, obviously vagus nerve will not be activated. As a result, there will be no release in the acetylcholine, so no stimulation in the G cells, parietal cells, ECL cells will occur. Vagus nerve will not be activated. As a result, there will be no stimulus that will stimulate the cells like G cells, parietal cells, ECL cells. As a result, stops in the gastric acid secretion or production, or you can say decrease in the ACL secretion. Now the fourth type of pathway, that is autocrine pathway, where transforming growth factor alpha is involved. So if there is an increase in the HCl concentration or HCl secretion, it will activate the TGF alpha, and this TGF alpha will increases the mucin release, which will form a protective covering concomitantly that will decrease the release of or secretion of HCl. So this is the end of my lecture. If you like my video, please do like, comment, share and subscribe. Thank you for your attention.